So defects in the aquaporin gene, in fact, turn out to have significant, very significant clinical consequences. Children with mutation have been identified by our colleagues in the Netherlands. And these children have severe nephrogenic diabetes insipidus. They must drink 20 liters, basically 40 bottles the size of water each day to avoid dehydration. Acquired defects are very common, and they're found in two sorts of situations. Individuals that have excessive fluid retention, such as congestive heart failure, overexpress the ATP2 protein, and those who cannot concentrate have a deficiency of the ATP2 protein, including the bedwetting in small children. They outgrow it, but they don't make enough aquaporin 2. I'm going to skip rapidly through now other members of the family. I'll mention ATP0. This has another name. It was the MIP protein of Lenz. We call it 0 because it was known as a polypeptide, but the function was not known. And we now know through the work of Masato Yasui, shown here, that it's important in the biology of Lenz, and mutations cause cataracts in small children. So there's going to be a theme emerging. Each new member of the family, depending on where it's expressed, has a different kind of biology. ATP4 present in the astroglial empty in brain. Now our brains are very special because there's no internal structure. They reside within our crania. The crania are rigid, protective, but this protective rigidity is a problem if there's a brain injury sustained either through trauma, a stroke, because when part of the brain is damaged, it swells, compresses adjacent brain tissue. The ATP4 protein is localized at the end of these astroglial empty. They look like suction cups surrounding the surface of the brain capillary. And you can see that immunoglobulin is present at this membrane, but not this membrane. Very interesting distribution. Now that distribution predicted correctly that this protein must play a role in the manifestations of brain edema. Brain edema is oftentimes the great cause of morbidity following a stroke or head trauma. It's the swelling, not the initial injury, that causes the big damage. In a series of studies undertaken with their colleagues in Norway, led by Mahmoud Amiri Mokhadam, I should mention, Mahmoud started his scientific career as a refugee in a camp in Pakistan, adopted by the Norwegian Social Services. He's now a leading neuroscientist. He discovered that the mutations in the expression of aquaporin 4, or the localization of aquaporin 4, are beneficial during defined brain injury. If you look here, the normal mouse has a larger amount of edema or infarct after brain injury than the mutant mouse. The mutant mouse suffers from epilepsy, so it's not preferred to be aquaporin 4 null. On the other hand, if we could manipulate the expression of aquaporin 4 following brain injury, we might have a method for prevention and treatment of brain edema, which would be a very important advance. Stroke is the third leading cause of death in Europe and the United States. Most of the morbidity and mortality is due to brain edema. So another member of the family, ATP5, is expressed in secretory glands at the apical surface, just the center part of the gland. So here's the gland for a rat. Here's the ATP5 surface. This is the last membrane water process during the secretion of tears, sweat, and saliva. Now, a study undertaken by our colleagues from Denmark, we looked at an aquaporin 5 null mouse created by Emil Menani and Carissa Crane at the University of Cincinnati. And the mouse looks and feels pretty normal. It has normal appearing sweat glands in the skin, but by a special technique we're able to identify functional sweat glands because of the release of amylase, the starch, the altered starch along with the reactive iodine. So the blue dots represent functional sweat glands. And what you should see, that after pilocarpine injection, the normal mouse has many, many dozens of functional sweat glands. The null mouse has a few weakly functioning sweat glands. Now, mice live in cool nocturnal environments. They seek the shade during the hot hours of the day, out laboring in the sun. For us to sweat is oftentimes a matter of life or death. In 2003, Western Europe, I'm sure Russia as well, sustained the worst heat wave, the longest and most severe heat wave in recorded history. Paris recorded 40 degrees Celsius and above for 2 weeks consecutively. And the public health officials registered 15,000 unexpected deaths. In Italy, the same thing, 20,000 unexpected deaths. Unexpected because these were older individuals, individuals in their 60s and 70s. But when you get to be 64, as I am, that doesn't seem so old. What does this mean? Is there an aquaporin involved? And the answer is still unsure, but most likely this does represent the age-dependent decline in physiological function of the aquaporins. Many physiological pathways decline as we age. At age 64, I love to exercise, but I can't run as fast as when I was 18. My heart won't beat as fast. Can't move oxygen as fast. So there are a number of pathways that decline in efficiency. And it's our thought that the decline of the aquaporins, immunal concentration, sensation of thirst, and sweat may have contributed to the demise of these individuals. We'll have to wait to confirm that that may be true. Now I'm going to shift to talk briefly about the aquaglycerophores, the glycerol transporting homologs of the family. In this slide I borrowed from Big App from the Morris Perkman laboratories, juxtaposes the pore lining residues, the narrowest part of the channel of aquaporin, which are shaded in darker colors, and an aquaglycerophore, which are lightly shaded. And what you should see is, so here's the aqueous pore, here's an arginate conserved in the water channel and the glycerol transporter. Here's the cysteine, the mercury inhibitory site that Bob Macy identified. Notice it's replaced the glycerol channel by phenylalanine, a hydrophobic, bulky residue extending out into the pore. Here's a histidine, a partially positive charge in the water channel. It's replaced by glycine, a tiny residue here. So it's been resolved with great certainty by molecular dynamics simulations undertaken by Klaus Schulten and his team in Illinois, Helmut Gruppmüller and his team in Germany, that there's motion for glycerol transport. If you imagine this phenylalanine as a hydrophobic trapdoor suddenly springing open, notice the glycine is very small. The pore is much larger, allowing the formation of a three-carbon polyol. So science makes sense. It's amazing. What does glycerol transport have to do with normal health or disease? Johan Oberyn from the University of Uppsala spent a year with us as a sabbatical worker, made this very nice preparation which shows immunostaining of aquaglycerophore 3 in rodent skin. Here we have prenatal mouse, and the basal levels are against staining. And here's an adult mouse, again, the basal levels is stained. This appears to be the glycerol permeated part of the skin, which is essential for skin integrity. Now, it didn't take long for the beauty industry to get interested in this. It was a few summers back, I was visited by some executives from the Christian Dior company. And I don't know about your laboratories here at Moscow State University, but I don't get many visitors to Christian Dior. And they didn't waste their time coming to Baltimore just to say hello. They invited me to Paris to give a lecture, a very nice lecture, almost as nice as this. And of course, they had a commercial idea. Their chemists had identified a small naturally occurring substance that leads to an upregulation, an increase in the aquaglycerophore expression in the skin, particularly sun-exposed skin, which they were offering as a beauty product. Well, I should point out, I have no commercial ties to Christian Dior. Some days I would love to have commercial ties to Christian Dior, but none were offered under terms I could accept. But the notion is, if you use their product, this hydroxy skin, it costs a lot, 50 euros for a 50-gram jar. If you use it on this, you'll look like this. I'm not convinced personally, but the beauty industry doesn't have to show proof of concept. Now, this was the back page of the Marie Claire Beauty magazine, which I purloined from the Charles de Gaulle business hours. And for those of you who can read French, look carefully. It talks about profound hydration, spectacular results, and what's this? The Nobel Prize in Chemistry. I showed this to my mother. She's not a university graduate. She's a farm girl, now in her 80s. I showed this to her. She was back in Minnesota, and she smiled and said, "Peter, I think you're finally doing something useful." Well, our mothers know us better than we know ourselves. And in fact, it is important that we should do something useful. And that's our object. Turns out that alpha-glycerol poetry is also present in red blood cells, conferring glycerol transport, which turns out to be very important during the clinical manifestations of malaria. And I now work as a director of the Malaria Research Institute. I spent a third of my year in sub-Saharan Africa. Malaria is my major focus. And you think for a moment, the parasite, during the red cell phase, will live in a vacuum within a cell, will suddenly grow and divide to 30 new daughter cells. How does it synthesize the glycerol lipids without the biochemical machinery? The answer is glycerol is transported across the plasma membrane, the vacuole membrane, the parasite membrane through aquaglycerophorate. And in a series of knockout experiments, we confirmed that the glycerol transport is diminished and the virulence of the infection is diminished. First, let me show you a picture of some real blood. So here's what I'm talking about. An individual parasite in a ring state will grow and divide to 32 daughter cells, eating all of the hemoglobin of the cell for fuel. And when this cell breaks, releasing 32 daughter cells, the cycle begins again. 2 more days, 32 times 32 is 1,000. Times 32 times 32 is 1,000,000. So in 6 cycles, we have 1 billion parasites releasing toxic waste, leading to horrific fevers and organ damage. So the knockouts, as I mentioned, will diminish the infection, will prolong survival, but they don't cure the disease. So here are normal mice infected with a wild-type malaria parasite. 3 weeks, they're all dead. The mutants live longer. 4 weeks, they're all dead. So it looks better, but not enough to be a drug target. I'm sorry, but that's research. You ask a question, you assess the situation as well and honestly as you can, and you either move on or move out.